Now, I guess we can get started. So let me switch to the proper view and say, yeah, thanks everybody for joining this call today. This is the 8th annual NABat Kickoff in BC here. We are, I guess, Wildlife Conservation Society of Canada. We've been coordinating the program in BC since its start in 2016. Just a quick outline of what we'll be talking about today. You may recognize this from some previous years, very similar structure. We'll be talking about the introduction, or talking about the NABat introduction of methods, going through our previous year's accomplishments and continuity, the submission and analysis logistics, talking about some of the paperwork for people who are on contract, as well as reminders and updates to make sure the year runs smoothly. Start with first. Our WCSC BAT team here, we have 6 members, starting with Dana Blouin, Heather Gates, Corey Lawson, Aaron Lowe, Brian Patterson, and then me, Jason Ray. There is, of course, other partners here, and I want to mention the significant involvement from Mandy Kellner with the BC Ministry of Water, Land and Resource Stewardship, who also helps coordinate the BC Parks and BC Ministry grid cells. NABat in BC is also guided by 2 main groups. We have met fairly regularly with our small provincial guidance group and a larger hub steering committee, which we will be convening more regularly in this next year following the formation of our new hub. We'll talk about that in a second, though. Names here are just organized alphabetically. So with that, I'll turn it over to Corey for an update on bat health and NABat program as a whole.
Okay, thanks, Jason. So yeah, just a few announcements, I guess, that are related to, but tangential maybe to NABat per se, and that starts with the State of the Bats report. This report was originally envisioned by the group that I worked with starting in 2015 to get the NABat program launched. And in the end, this took a different kind of face than we thought it would, with Bat Conservation International actually producing this report rather than the NABat committee itself. So what kind of evolved is the NABat committee, which is out of Fort Collins as a head, the USGS then is running the program, and they're doing all of the analyses, all of the underpinning stuff, a lot of on the ground and actual analytical stuff that then needs to feed up into these communication products. So this year there was an NABat, well actually last year there was an NABat stationary acoustic report, and then there is a transect one that's going to be coming out very shortly. And in the interim, there's this bigger State of the Bats report, which included some of the NABat results, but much larger as well. And this was spearheaded by NABCA. If you've not heard about that, that's North American Bat Conservation Alliance. And that was all part of this kind of bigger North American bat monitoring idea as well. Get the NABCA running, which would oversee all the kind of the working groups in North America for bats. And as part of that, then they put together the State of the Bat report with BCI. And so if you just go into, well, I guess you can kind of see the address at the bottom, but it actually, if you just type in stateofthebats.org, it will take you to this as well. And kind of the key highlights here, this is a lot of expert review that's gone into this as well, is about almost 50% of the bats in North America have some sort of conservation concern and need conservation attention. And so if you're interested in more of that, I encourage you to look at that report. They've got a lot of great stuff, including projecting what's going to happen with things like climate change and wind energy, for example. And of course, as you can imagine, some dire outlooks for that. Okay, next slide, please. Okay, so other bat news, I'm sure you guys will have all heard this, but just the PD spread update. Corey Olson and Susan Holroyd, who basically run the Alberta component of WCS Canada's BAT program, is kind of evolving more to be a prairie program. And Corey led a bridges project where he was sampling and having a lot of people sample hundreds of bridges across the prairies. And so Saskatchewan, that's how PD was discovered first in Saskatchewan 2 years ago, then last year in Alberta, again, we're talking bridges. And then here in British Columbia, Mandy Keller, the provincial government, spearheaded getting about 100 bridges sampled in this province. And one of those in the Grand Forks area tested conclusive for PD. And so now we do kind of officially have PD basically from coast to coast in the country. And next slide. Okay, and then stemming from kind of, well, I guess both of these threats really in a way, looking at wind energy in particular, and maybe a little bit understanding potential PD impacts, comes this Kasiwik assessment. The Kasiwik report on the 3 migratory species was written for Kasiwik and then finally reviewed at the, well, I guess last month, the end of April then. The assessment came forward from that group saying that these 3 migratory species should be endangered. The presence of PD being found on silver-haired bats, but the uncertainty of how or if that will impact that species did apparently have some play in the listing of that species. And then for hoary bat, really they were using and pointing to the models. There's a couple of published papers on models with some very dire outlooks, including by 2028, that species being 50% reduced in population. So this is all kind of, ramping up to obviously highlight the importance of NABat monitoring, and that we are very lucky that we're sitting basically with 8 years now, because kind of the magic number we're being told is 10. We got a pretty solid baseline upon which to gauge trends, and so we're almost there, and in good timing, because obviously these things now are expected to hit the bats harder. Yeah, you can go on. Thanks, Jason. Okay, so kind of the last thing then, pretty obvious, I think, but in case you're kind of wondering where the grid cells fit in, obviously each grid cell is a component of a broader regional analysis, and then that rolls up into a provincial analysis. And what's coming online now moving forward is actually rolling up into kind of a more Alaska, BC roll up, i.e. kind of northwest of North America. And then ultimately our data feed into the continental program. And there's a lot of products that are starting to appear, and a lot more to come, especially as we move into different types of data streams. So I think that leads to the next slide. Okay. Oh, actually, no. Okay. But on this same vein, nabatmonitoring.org, if you go into that website, you can see all of the products that are coming out. Those of you who have not had a chance to look in that website, it might be interesting just to take a look at what it is you're feeding into, because there's a lot of stuff that's coming out of this. Next slide. Okay, including then the status and trends. So there's some, expected huge declines. Yeah, you can just put those, yeah, you can just continue with those. Jason, thanks. So for the most part, we've been focusing on acoustics, but NABat is much larger than that, and we are gradually increasing the data streams that feed into understanding the state of the bats. So we've been focusing on summer abundance largely with transects, and then also looking at passive data in terms of species ranges and abundance. The BC Community Bat Program has been doing a lot of colony counts, and the idea moving forward is to hopefully be able to pull these data streams together, and certainly you'll see on the field forms that there is more information being requested on known colonies in or around your grid cell. The idea then would be eventually to use, even at the continental scale then, summer occupancy, winter colony counts, summer colony counts, and transects. And they're already starting to do that. They've actually been using known winter locations and trends of populations at these winter locations to actually inform summer abundance models. And so it's gotten pretty complex. And there's a pretty cool team of statisticians working on that in the Fort Collins USGS office. So suffice to say, the more data you can gather, the stronger the analyses can be, and it doesn't stop at acoustics. So just putting the plug in there for doing as much as you can, even beyond the acoustic monitoring that we're doing right now. Next slide. Okay, yeah, and I think this was last year's. Basically, you can go in there and see the trends for the species that did have enough data to calculate those trends. And the next slide. Grid cells in North America, you will notice this is just US and Canada. Mexico actually has their own monitoring program. It looks like a lot of grid cells. This is basically anything that's being done in North America for grid cells at this point. That does not necessarily mean all acoustics. Some of these, come online for various reasons, maybe hibernacula counts and so on. And so, yeah, BC, I mean, we're a large province and we're getting there, right? You can see, we've got a good scattering of dots. Be lovely to get more over time. But for now, I think, we've done a really good job at covering fairly well. So you can see then kind of the dispersion of the grid cells, obviously a lot in the south. We hope to get more in the north over time. Nice that we're focusing on the south, given the white nose spread. But in the northwest, you can see there's basically none, which is why we were so excited that Southeast Alaska came to the plate and said we would like to coordinate with you guys. And so now Jason will be mentioning a little bit later about the Southeast Alaska hub that has grid cells, so that even though we can't really easily monitor the northwest part of BC, it's pretty much unroaded, as most of you probably know, we will be able to extrapolate what's going on with our bats, hopefully based on the data we'll get from Southeast Alaska. I think that's it.
That's good. Thank you, Corey. So yeah, with that, basically I'll focus in a little bit more about BC. In BC, we have 10,124 potential grid cells, 10 by 10 kilometers each, and currently we monitor just over 60 grid cells. In 2023, we're looking at, I think it is 62 is forecast right now, but we'll see. We're still adding in a couple more over time. And in each of these grid cells, we're using acoustic recorders to capture ultrasound the bats use to navigate their environment and identify the species present in the grid cell from that. As Corey said, we were focused on establishing our grid cells in southern BC initially, but we're expanding further northward now as opportunities present themselves. The goals of our monitoring in BC, we're looking to establish bat diversity and relative abundance and activity, provide the architecture for coordinated bat monitoring to support local, regional, and range-wide inferences about trends in bat population and abundances in response to white nose syndrome, climate change, wind energy development, and habitat loss, or other threats. Quantify the species-specific impacts of white nose syndrome and or other cumulative stressors by detecting and measuring changes in bat activity and occurrence. And finally, provide a quantitative method of evaluating mitigation activities, recovery actions, or other management and conservation efforts in the areas being monitored. So those are the big goals that we're using your data for. So very briefly, because you're all very super familiar with our procedures, within each grid cell, you deploy 2 to 4 stationary detectors, and these are SM2s, SM4s, or NABat Swifts for 7 nights per year. I just want to take a second to recognize the photos and deployment locations. I love seeing these photos personally. They are also invaluable for establishing the history of the site, noting any changes between years, deployment and local site conditions, whether trees have grown or been cleared, buildings built, forests chopped down or burnt down. And we also use these photos for sending to funders and using in reports or social media, things like that. So please keep sending them in each year. Also, please include people in them wherever you can. It does make these photos more engaging, but we also need a photo release if you have people in your forms. So this current location is one of our new grid cells. It is Naikun on Haida Gwaii, the very western end of our province here. This one is Elko. It's a nice deployment location, very open, habitat quite a bit of roosting habitat around, as well as a nice linear feature following the forest edge for the bats to follow. Similarly, this is one of our detectors in our Vernon grid cell, also a nice linear feature along the road to follow, and a very, very open habitat that's unlikely to be filled in anytime soon. Another detector on Vancouver Island. This is near the Goldstream, or this is in the Goldstream grid cell near Victoria. This one is a forest clearing with quite a nice opening around and catches relatively few bats, but very nice recordings as it's all quite open. And last, I usually like to throw Pemberton here because I really like this deployment location. It's got a nice flyaway potential for the bats commuting along the bluff edge over the river, as well as following the river itself. And it's also just a very, very pretty location. So like I said, please do keep sending in those photos. If you do send them in, we would actually ask that you also send in a photo release form that gives us better confidence that we can use those photos freely. On the flip side, if you don't want us using your photos, please let us know. I'll tag those photos as data use only, and we won't send them around. So these photo forms you see on the screen, this image there, this is available on our Google Drive. It can be printed out, signed, and then sent to Dana Blouin, who is listed on the bottom of the form. So while your stationary detectors are out in the landscape, you're also being asked to collect 2 driving transects in most grid cells. So as you know, bat detector is mounted. The bat detector microphone is mounted to the car roof. You drive your car at 30 kilometers per hour for roughly an hour to an hour and a half, which it ends up being 30 to 45 kilometers. This specific speed is chosen because it is typically faster than the average bat would fly, so we can actually assume that each individual recording is a different individual bat and allows us to estimate relative abundance from that. Now, for both transects and stationary deployments, consistency is absolutely critical between years. This means making sure to keep the specifics of your deployment and data collection the same as previous years. Things like pole placement, microphone orientation, which exact microphone and detector you use, as identified by serial number, should all be kept the same if possible. So maximizing that consistency means more precise trends in the end, better data. Another reason for filling out the field forms too is keeping track of this consistency, knowing which microphone you used last year, telling us which microphone you used each year as well, giving us the confidence that everything is the same. We will get to those field forms later though. So there are, of course, some reasons that are unavoidable for shifting detectors, though. I want to mention that now, too. If you are unable to monitor the same spots or you need to shift your detector location by X meters or even to a different site, please just let us know. That does happen from time to time, and it is a scenario we are able to accommodate. Okay, and we are typically keeping our stationary and transect data roughly separate here, and they follow 2 different pipelines. So your stationary data gets analyzed and turned into a model up here, as well as the transect is analyzed and turned into a paired model as well. So stationary data collects recordings over a much broader period of time, and those detectors ultimately detect recordings from most species in each grid cell each year. Whereas the transects are only about 2 to 3 hours of observation per year, so they collect fewer recordings and fewer species, but they give us that critical element of being able to assume that each individual recording is a different bat. Now, both stationary and transect detectors do record different individuals because the transects are covering a different geographic area. So we can actually use the trends from both of these to compare to one another, and agreement between the 2 does provide a sense of validation in our trends between both of them. And just to note, the Continental program is using the transects for abundance and stationary only for occupancy models, but we're using both. We're using stationary for activity estimates as well. So we're using models on the changes in activity between years. So a slightly different procedure, which we're hoping will give us a more sensitive metric of this changes in relative abundance in the end. Now, we can't talk about activity without mentioning covariates. I've just included some quick icons for all the covariates we've considered so far. That activity is of course heavily influenced by the covariates of the landscape, and they change frequently throughout the year and the night. Temperature is an easy one that I'll call out here. Lower temperature means insect activity declines, and thus bat activity also declines. That 10-degree threshold that you should remember from the transect guidance comes from this relationship. In BC, we suspect the insects should not be flying, typically aren't flying lower than 10 degrees Celsius, so the bats likely wouldn't be either. In our models so far, we have included temperature, humidity, precipitation, wind speed, moon phase, and in 2023, we added degree days and total accumulated precipitation covariates. The influence of each of these will, of course, differ by species, and future analyses will include that, as well as the potential for interaction between these covariates. So how moon phase might be influenced by precipitation and humidity. For instance, if you can't see the moon, the effect of the light from the moon will be very minimal. Or vice versa, if the moon is full and there is no cloud cover, the effect should be stronger. So one of the temperature covariate that I've focused on so far is collected in grid cell. This is one of the most important covariates we have. And your grid cell temperature logging typically looks like this. Now, we can fill in some of the temperature and humidity covariate from nearby weather stations, but these direct observations from within your grid cell are still our gold standard because they are collected at the exact same elevation, the exact same sun exposure, things like that. So this will be likely more precise than what we're getting from a weather station that may be within 5 or 6 or 7 or 8 kilometers away, kind of thing. Inside that ambient shield, your actual logger device will look like one of these three. We are currently sending out Bluetooth Kestrel drops as well as Bluetooth hobos. If you have some old equipment, you may or may not still have some optical hobos out there, and that's the one in the bottom left. The Kestrel obviously being the one on the bottom top right, and the bottom right is the Bluetooth hobo. Now, the Kestrel is just shoved up inside or dangled up inside the ambient shield with a string. It is tied to the top of the plastic container itself, and you just kind of release the twist tie, lower it down, and this green Kestrel drops out. You can see, let's see if I can put a little image or something on here. Push the laser pointer. You can see this little button here is the test light. Push that to see this LED blink, and that'll tell you that your device is still functioning properly. If you push that test button and the LED doesn't blink, you'll need a new coin cell battery. Just pops in the back. It is a CR2302 type. And note, if you've removed the battery or the battery has died in this device, you will need to reconnect to your phone so it can pull the correct date and time from your phone. Otherwise, it will be collecting temperature recordings from time 0, which I think is January 1st, 2000 on this model. The other type is the Bluetooth hobo. That inside of that ambient shield will look like this. Similar kind of test procedure. You would push this button here to test, and you should see these lights blink. This one also connects to your phone or tablet or other Bluetooth-enabled device. And we have a procedure paired for both of these actually, on the Google Drive. So if you're not 100% sure how to use these, just check the Google Drive for those procedures. But basically, yeah, push the test LED, watch for the blinking, and connect to your phone to download the data and send to us. The phone app is called Hobo Connect, I believe. And Aaron has just written a new updated procedure for the recent changes they made to that app. Okay, and I guess one last thing to note about this is if you're receiving this device from one of the WCSC kits, the batteries should have been changed. But if you do find it dead, if for any reason, it will likely be a mechanical failure. But otherwise, let us know and we can send you a replacement AA 2/3 size lithium battery. The last type of logger is this optical hobo. There should be very few of these circulating, if any at all, but they will need to be read with a paired optical reader that attaches to the end here. So if you have these and you're not familiar with how to use them, please let me know. Otherwise, please do just send these back at the end of the observation period, because we will need to pull these old devices back in and download the data from them. New in 2022, we added a slightly more specific temperature measurement along the transects. We have a 5th hobo in all kits to attach to the car mount microphone and measure the temperature every minute along your transect. The idea being that this gives us a better, more precise estimate of temperature and humidity along the transect as your car goes up and down valleys, and temperature can change by 10, 15 degrees even on certain nights and certain locations. So we're capturing temperature and humidity changes along the entire transect, using that to be a little bit more precise of a covariate for your transect activity. I'll pass to Aaron for the next slide.
Great. So also new last year was using the sticky traps to capture insects, just to get an idea of the metric of insect density along the driving transects. So same as last year, we're asking people to mount the sticky traps on their hood, but re-emphasizing this year, just sticking those sticky traps as close to the front of the vehicle as possible would be great. Yeah, and then once the transect is complete, you're going to pull that insect sheet off. We've provided sheet protectors this year instead of the saran wrap, so it's just to make the covering a little bit more smooth, to help with how we're analyzing it. So you'll put that sticky trap inside of that sheet protector, and then also, rather than writing on the back of the sticky trap, we're asking that you write using a permanent marker just on the actual sheet protector itself. So how I have the photo just at the bottom. So your grid cell ID, your name, the date, as well as the transect number, and just making sure all of that information gets written on there. Otherwise, we're not able to kind of correlate it with where it goes. Once you have all of that, the sticky traps are going to be mailed back as well. So in your kits, you're going to have a prepaid and pre-addressed bubble envelope that you're going to put the SD cards on, which I think Jason will talk about later. So the SD cards will go in there, and then you'll also mail back the sticky traps kind of once you've completed your survey. Yeah, and just taking pictures as well. So taking pictures of data sheets, taking pictures of the insect sticky traps, and then it just kind of provides us as a backup in case things go missing. Yeah, I think
Perfect.
that's everything.
Perfect, thank you. All right, so next I'll move on to the next section of accomplishments and continuity. As we've said before, this is now the 8th year of monitoring in 2023. Our total data set will contain, or does contain, over 1.25 million bat files. And this year we expect to pass the 1.5 million bat file mark. And it's great to see so many people who've been with us from the very beginning, combined with so many dedicated newcomers along the way. Our data set is one of the larger ones in the program because of this. Now, each year we've detected the presence of all 15 species known to be in BC and provided additional acoustic records of 2 southern species, Canyon bat and Mexican free-tailed bat, in certain grid cells. Interesting results from past years of monitoring. We have expanded known ranges for 6 species and filled in acoustic detections for new areas for nearly all species in BC. I'd still like to highlight the eastern red bat example, as this species has continued to expand through new grid cells in the province. And these range maps that I'll be talking about have been incorporated into the newest BC CDC range maps, as well as the BC Bats book from Corey. You may remember also we expanded the western small-footed and fringed bat ranges in 2021, but we have once again gathered new records from these species again and expanded the ranges slightly farther outside expected range in 2022 as well. I think western small-footed, I believe, was in the Creston range, as well as the fringed Metis popping out further in this grid cell, the Kimberley grid cell as well. So we continue to update our activity trends this past year as well. We've included additional covariates like the degree days and total accumulated precipitation that I mentioned earlier. We've eliminated some outliers as well as refined the models themselves slightly. In 2023, new changes to the NABat software we use will provide the option to highlight feeding activity in our recordings. So we'll be looking at incorporating this new tool into our workflow to rank or weight sites by proportion of feeding activity. We will also once again be developing slightly more sophisticated approaches to specific species specific and regional models as well that will allow for variable effects of covariates and interactions between covariates. So after considering species identifiability, detectability, and number of files, we're able to calculate trends for 10 species here. We identified no significant trends at a provincial level from using our auto ID data though. The absence of this trend in big brown activity is notable difference from last year. We are still gathering enough data to smooth out the annual variability in activity we can see across our sites, as that trend came with primarily 5 and 6 years of data, in other words, few data points. But we'll still be examining that species in particular in the future as a species of interest. And this year also of note, we did recalculate the power to detect changes in populations with our current sampling design and reinforce the importance of gathering a minimum of 10 years of data. Thus, as you'd expect, all these monitoring sites represent long-term and extremely valuable investments. You donate your efforts each year to assemble this massive baseline, and each year the value of the dataset increases. And this is especially notable as this past year, we just passed the 5-year mark for nearly 45 to 50 of our grid cells, which is huge because 5 years is the minimum amount that we said we could start estimating trends with. And once we get to 10, we'll start to have much more reliable numbers from those. Now, solidifying these long-term monitoring sites and fostering involvement for the duration of the program is one of the major goals going forward. But we do still consider expanding to new grid cells opportunistically in key areas. For instance, we've added three new grid cells for northeastern BC, as well as the new grid cells we're adding into our program for the hub collaboration. So to date, we've spent a huge amount of effort establishing the program, preparing procedures, workflows, and tools to establish efficient operations. WCSC's guiding mission, though, is to gather research and use this research to address conservation issues, and coordinating ongoing monitoring program does fall outside our scope for continued implementation. So with the establishment of our new BC Southeast Alaska hub, we are looking to step back and take a secondary role in the organization as another group can take the reins to lead the monitoring through BC and Southeast Alaska in future years. This transition will take a little bit of time as we find organization to take over. And Corey, did you want to jump in and say anything on that one too?
Sorry, I just had to unmute. Yeah, no, I don't know that we have too much to say at this point. We're kind of early in the process, but we are working with the BC government and the Alaska government to talk about a plan for having a more centralized hub coordinator, and that would lie outside of WCS Canada. But yeah, we're basically working over the next year to try to make that sort of a seamless handoff, and looking forward to still supporting this initiative. We know it's very important. That's why we started it. But as Jason said, we are now feeling like we can hopefully hand over kind of a well-organized program on a silver platter, if you will, and hopefully that will continue, and we will kind of work on going back to some of the more fundamental research and knowledge gaps that we know still exist in the province. Now that we'd be looking at kind of wrapping up the coordination of NABat as one of our major projects to date.
Sounds good. Thank you. Okay, so next we'll move on to the submission and analysis logistics. I'll pass it back to Erin.
Sorry. So data submission will work the same as it had last year. So each grid leader will collect and receive their equipment. You'll then deploy it. You'll retrieve the data and then you organize it and send it to WCS, basically in 3 different steps. So basically, we're looking for triplicates of the data. So first, once you've received or collected your acoustic detector, you're going to copy all of the data from the SD cards to your computer, and then also to the USBs that have been provided in the kit. So that's that 3 kind of versions of the data. Once you have the data on your hard drive and the USB, you're then going to label the SD cards with the quadrant and then the grid cell ID and mail it to us. And as I mentioned before, we're also asking for the sticky traps back, right? So it'd be the 2 SD cards as well as the sticky traps, those 2 sticky traps sending back to us in that prepaid envelope. So sending us that back as soon as possible. You will then organize the data that you've pulled off of the SD cards into the folders, and we've given in the protocol document kind of that how we'd like it organized. So organizing those data into those folders, and then you're going to add the site photos, the field forms, the temperature records, all of that to kind of just organize nicely on the USB sticks, and then placing that USB kit back into the kit and then sending it on to whether it's us or the next grid leader. So we're asking that, as I mentioned before, that we're taking pictures of all the data sheets as well as the insect traps, and all of that's going to live on the USB as well, just in case something goes missing. We have that to go back to. Yeah, so everything, we're hoping, you know, people are efficient with passing it along, but having everything back to us submitted by the end of the monitoring period, so by August 1st. As I mentioned, yeah, you'll have the pre-addressed and prepaid bubble mailers that you're using for the SD cards and the sticky traps. And then once we receive the data, we'll be running everything through Kaleidoscope and NABat, and then sending it to Brian for him to kind of take over the next steps. Also, as it was last year as well, there's going to be that Google tracking sheet. And this is, we're emphasizing that this is used this year, just so that we have an idea of when things are moving along. Otherwise, we'll be getting emails from me just to kind of make sure that things are making its way along, and we know where equipment is. Yeah, and then just procedure documents, guides, fields, forms, all of this is available on the Google Drive site, and so you can reference any of that at any time, and Jason will share that link after this meeting as well. So
perfect.
Yeah.
Thank you, Erin. Can somebody else in the CASLA office copy and paste that link to the spreadsheet into the chat, please? Because I'm not able to get it from the presentation, it seems, right now. The presenter notes, rather.
Yeah, we can do that.
Perfect. Thank you. So I'll pass it over to Brian next for the latter half of this section.
All right, thanks a lot, Jason, and everyone there. Yeah, let's see here. So I'd like to reiterate the importance of you know, just having that organization in the field and trying to fill out the you know, all the field forms and whatnot as while you're out there. Also with, you know, saving your all the data, saving it in the appropriate format that'll be available for you in that link. So that's the appropriate folder and file structure there. It just saves a lot of it makes it a lot easier, I think, for everybody. When it gets to WCSC, it makes it a lot easier for them to organize it. You know, it may take you an extra 5-10 minutes to organize it in the right structure, but you can imagine doing it for 50 or 60 cells. It can get confusing and take quite a bit of time. And similarly, when I get it, if it's in that nice format, and I'm analyzing or classifying, I should say classifying, I suppose, all the BAT passes, all the files that we get. And if it's in the appropriate, you know, folder structure, then it's a lot easier for me to sort on my end in the programs that I use to do that with. And, you know, then I don't have to, if it's not, sometimes you may get a big jumble of files that aren't necessary. I may have to play with it a little bit more in order to basically get it to a nice format where I can easily work with it. So as Erin was saying, we run it through 2 forms of auto identification. That's the Kaleidoscope Pro and the NABat. and as you're maintaining 3 copies, we're also maintaining 3 copies, just in case anything was to ever happen. We've got basically a raw folder that's the data as you've sent it essentially. We've got a process folder which has the auto ID'd information, and then there's the analyze folder which has the manual verification of those. And that manual verification, there's a process that I think it may be available on the through the Google Drive, but it's standardized for what we're doing here. And yeah, it can definitely take some time. Some grid cells are quite quick when there's not a lot of files, and then other ones there can be a ton of files and take days to go through. And of course some of those have are nice and clean, and some of them have multiple bats in every one, and sometimes they're high frequency mixed with low frequency and whatnot. So it can take a little bit to sort those things out. And then when I'm finished with the data, it takes me quite a while, but then I'll send it over to Jason. Everything's backed up, and he basically uses scripting algorithms and such that he's developed to, I guess, facilitate the information that gets spit out and makes it easier to share with the project partners. And of course, any delay to this process, is further delay on our end as well. If I'm not getting the files in a timely fashion, then it's, I'm not able to necessarily analyze them in a timely fashion. So let's go to the next slide, please. Here's just a couple examples of some bat call sequences. This is a silver-haired bat, as we can see by the nice flat calls around, or pulses, I guess, around 25 kilohertz. So that would be pretty easy. Next one. I think this is an example of a big brown bat. It could be easily confused with the silver-haired bat in this situation, both kind of in that 25 kilohertz or lower range. If it gets low enough, though, you can say it's a big brown bat. Go to the next one. This would be a little brown bat. It'd be, it's probably a little bit hard to tell from this one, as to often manually I'd look at slope for that. But the process is you use those 2 auto ID programs. If they, if they both agree and you've got a nice clean call and you really can't dispute that, then we'll accept that. We'll accept that auto identification. Next one, please. Here we've got an Eastern red bat, and depending on how you expand the file, you could see that there's an undulation in here, and there's even an upturned toe, sometimes dialing that gain back, or the full spectrum back a little bit, can sometimes help you to pull those things out. But that upturned toe and undulation is something that we look for in Eastern red bat. Through the auto ID process, if Eastern red bat's expected to be present, we get a lot of Eastern red bats auto ID'd, and they often aren't, necessarily Eastern red bat. So we do have to be that's one of the species we have to be particularly careful about. Next, please.
And a reason why manual ID is super valuable too.
Yeah, there's a lot of situations where, I could go on and on about the situations where the auto ID sort of falls down, but it does provide, it really does, though, provide a way to standardize a lot of the files that, are really hard to consistently pull out the same designations. And that's what we're kind of looking for in this program. This here is a Myotis evotis, or long-eared bat. Usually a pretty big sweep to the frequency, and it'll go down to 30 kilohertz. There are times where you could confuse it with a big brown bat, for instance. And so you do have to be a little bit careful there, but if you look at enough of them, you kind of figure it out. And I think the new Bats of BC book that came out recently, that I think Corey had a part in there. There's a lot of great information on that. So if anybody's interested in Bat ID, reach out to any one of us. Feel free to reach out to me as well. And Corey's book is a great reference for that. It can be quite challenging, but the more you look at it and the more you see, the more you're able to do it. Here, I've got my bar in the way, but I think that means it's so low. It'd be a Yuma bat here, or not Yuma, sorry. Yuma is EUMA, so spotted bat. But yeah, that's really the only species in BC that's audible by us humans, and it can be down around that 10 kilohertz range. There's certain parts of the product. It's always cool to pick them up. One of the challenges, I mean, in my idea, is that that's quite an expanded. If you look at the axis on the left side there, that's quite an expanded axis. So not necessarily always looking at all my grid cells in that expanded axis. So it's really important to especially where you expect to have spotted bats, to open it up, to run filters that are going to pull it out and so on. So just things to, and then there's noise. There's insects that'll get down there too that, you have to tease out as well. Next, please. And here you can see we've got a situation with multiple bats. We've got one that's, around that 40 kilohertz mark. So it's a myotis of some type. Then we've got another that's kind of around that 25 kilohertz, and there's a feeding buzz in there. So that would probably be a silver-haired big brown combo. So those are things that do take a little bit of time to tease out. And what you could have happening in your auto identification is that sometimes one program might pick out one and the other might pick out the other, or Sonobat might just simply give you a high-low. And then, it's kind of up to me to pull those out again on my end and try to separate them as best I can. Next, please. Another challenge, if the detectors are not necessarily set up in a nice open area that's free of clutter and other features that could create echoes. So whether it's bluffs, water, whatever, is that you can get these sort of echoes that are happening, and they can confound the auto ID. Definitely, they can also make it challenging for manual verification, but. Sometimes you can pull species out of them as well. It does make it harder to say run one of our filters, one of the filters that we've developed to help us pull out, further pull out species when we're doing the manual verification. It sometimes may get tripped up on some of those echoes and whatnot, or want to consider it as something else. So there's various, tricks you can do to clean it up a little bit and so on, but there's really no substitute for having someone's eyes on these things, or a lot would get missed. Hoary bat is another one that is often pulled out in the auto ID programs, but is not necessarily there. So it's just important to do it and to try to have the most accurate data set that we can manage. And that might be it for me. Is it, Jason?
Yes, perfect. Thanks very much, Brian.
Okay, thanks.
With that, I will pass it on to Dana for the paperwork section.
Okay, hello everybody. So I'm the one that has to give you the bad news, which is to say that there is some paperwork involved in the NABat program. Most of you have seen or spoken to me over email, so I look after all the contracts and invoicing for WCS Canada here. We do have a number of folks that we do contracts with to deliver their NABat grid cells. So just to say that these will be coming your way within the next week. Hopefully that is the plan. We ask that you take some time to read your contract. There's actually 2 places to sign on your contract. Important deliverables and reminders for those deliverables will be sent via an online contract safe program, it's called, that WCS uses to actually manage all of our contracts. So just to say that you'll be getting these automated emails throughout the year reminding you of your deliverable deadlines. And just to watch for those emails and perhaps check your spam filters, because sometimes they get filtered out. We ask that you submit invoices at the end of your deployment period. You're welcome to use the WCS invoice template, which is provided on that on the Google link that Jason has set up. And we also ask that you itemize the GST on your invoice. So this includes even your supply purchases, like batteries, for example. Standard for WCS Canada, we use the mileage rate per the National Joint Council rates, which is currently 0.545 per kilometer. And unfortunately, WCS Canada's year end is June 30th, which means right smack dab in the middle of the NABat season, which means I need invoices for any work that happens before the end of June, within the first few days of July. And then anything that happens after July 1st, we can get those invoices later. But yeah, and this part is quite important, and just as far as reporting on and tracking for all the plethora of grants that we managed to actually make the NABat program happen for the province. Next slide, Jason. So actually, Erin has already talked about this a fair bit. So there's the link right there for the Google Master Sheet for all the logistics. And I had just a quick note here on equipment management. Heather's going to talk a lot about equipment here very shortly. But again, just to say, you might get automated messages from Snipe IT, which is our equipment inventory system. Also, please don't ignore those. All this equipment that is lent to you by WCS Canada is needed elsewhere after the NABat season is completed. So yeah, that's it. Thank you.
Cool. Thanks, Dana. And last section, we'll pass it over to Heather for the...
Okay. So batteries, as we did last year, we are not shipping out batteries with the exception of any grid cells that are sampling at 384. We are sending out ProCell Ds in that case. Otherwise, please buy your own batteries and make sure you keep your receipts and submit them for reimbursement. Also, please don't ship batteries back to us. You can use them if they're not dead or recycle them locally, whatever. If the kit's being sent back in a vehicle, then feel free to send them back and we can deal with them at our end. Next slide, please. Okay, so yes, double check everything. I have set them, but that doesn't mean I haven't made a mistake somewhere. So set your prefix, leave whatever's there. It'll say CLL1 or AZ5 or whatever. So leave that and then follow that with your grid cell number and your quadrant. Yes, so as it says, they're all set to -7. If you're in Mountain Time Zone, please change that to -6. Make sure your lat long are set correctly. There will be numbers in there. That doesn't mean they're the right ones for your grid cell. Yeah, so the gain, the switches are set to 12 decibels on the SM2s. And yeah, I think, is that about it for that one? Yeah. Yeah, so please be gentle with these. They're getting old and they get damaged quite easily. So grip them at that black base when you're pulling them off. Don't hold on to the silver part and pull because they will pull apart at the base. Yeah. And next. Yeah, don't do that. That makes me very upset. I don't like dealing with that. Feel free to just reuse the wire if you want, recycle it, or send it back, but just don't send it back like that, please. Oh yeah, I receive a number of these back every year where everything has been ripped off the front of the detector. Everything's there for a reason, so please leave it. The Gorilla Tape over the LED is a little flap, so you can still look to make sure that it's working. And yeah, just gently open the Ziploc bag and everything should be able to be reused for several years.
Perfect. Thanks so much, Heather.
Okay, thanks.
I'll take over for the discussion of the field forms. As you will remember from last year, we updated our field forms a little bit. We have made some other small changes again this year to streamline them for hopefully the last time. So I'll just kind of go over the information here and just a reminder that these field forms are critical covariates to our analyses. They often inform the types of species we should be expecting in your grid cell, or in your location, the quality of calls we should be expecting, and if things don't match what we'd expect from these field forms, it gives us cause to go back and say, okay, so what did happen? So once I receive these field forms, they'll be coming to me in either Excel or Survey 123 submission, and I'll get to the Survey 123 thing in a few slides. But the Excel forms, I'm just reading into one main big data table through Python in the end. So transcribing those forms on your end and sending me the Excel is super helpful. But if you really don't have time, we are okay to take just like snapped images of your field forms too. It just means a little bit more effort on our part. And when we have 62 contributors all sending us images, that can be quite a lot of transcription time for one person. So I'll just kind of go through the fields so everybody has a sense of what's on these forms. Starting from top left is grid cell ID. This will be something you're hopefully quite familiar with, or otherwise you will see through communications with us. We'll be able to tell you which grid cell ID you're monitoring. This is also called GRTS ID in certain spots. Grid cell quadrant, basically just the direction in that 10 by 10 kilometer that your grid cell is placed within, the quarter of it rather. If you have moved your detector and we have, I guess, allowed you to do that, you will be changing the number on that quadrant. So from NW being northwest, it would then become NW2 if this is the second deployment location, or NW3 if we've moved it a couple times because we couldn't find a good ideal location for it originally, or maybe access was lost a couple times, so on and so forth. Next is the detector number of total deployed. This is saying I've deployed 3 detectors today elsewhere, or this is the 3rd detector I'm deploying today, more correctly, of 4 detectors I will deploy in this grid cell. So either or otherwise 2 of 4 or 1 of 4 or whatever else. Then we have location information down below. We're asking for this in decimal degrees now because we gave the option previously, but it didn't seem to be necessary. So we've just trimmed it down to only decimal degrees. And you can use NAD 83, WHS 84, whatever map datum your GPS prints out in, but please do send in the decimal degree location this time. Write your name, write the nickname for your grid cell or quadrant. This isn't necessarily super important, but it does help us match this up with the grid cell ID or other things to check for errors. And the grid cell quadrant, or sorry, rather site and quadrant name is something that you would be familiar with. You can call it maybe the road name or whatever else. The date that you started your recording, typically the date that you deployed. The date the recording ended, typically the day you retrieve the detector. Elevation of the site, as well as telling us how to get to the site. If you do decide to pass over the grid cell to somebody else in the end, we can then give this information to the next grid leader so they know where to go. The landowner and the contact information for the person who will give you access to that land. The same kind of thing. If you're no longer able to monitor, we use this information to pass it to the next grid leader who then contacts that person and gets access to the site. Next section, we have some information on detector details. So you'll be recording your detector ID and serial number, or rather detector ID or serial number. If you're using equipment from WCSC or the Parks detectors, they each have a nickname assigned to them and it's written somewhere on the device itself. So the serial number for this device ST2 would be something along the lines of like 03452 or whatever. But we've written in paint ST2 on the side, and that's that detector's nickname. So we prefer the nickname because it's easier for you to remember, and it is written in your files, as Heather was saying. It's in the prefix location there. So it's added to every single recording that comes out of that detector. That's why we ask for it in this field form. Similar kind of thing, we have the microphone serial number or microphone ID number. Same kind of thing. It has a long serial number or a inscribed little nickname on it. You can use either one. And both of these fields help both you and us ensure that you are deploying the same detectors and microphones in the same quadrants each year, because that will improve the consistency of recordings we're getting out of your efforts here. Otherwise, we're asking for bat detector model and microphone model. These should be fairly self-evident. They're just obviously the same detectors you're using last year, so it should be more or less just enter the same information. Microphone height in meters, and microphone orientation is going to be depending on your microphone type, but typically either 5 degrees below horizontal or completely vertical if you're using an SM4 bat detector. Next, we ask for the detector settings. This is oftentimes something that a lot of our grid leaders don't typically fill out, but we do still get benefit from having these settings listed because it tells us that you are using the correct settings that we've identified in the field form. These settings are not necessarily printed into the files, so oftentimes the only way we can be sure that you did use gain 12, for instance, is if you tell us right here. So please do go through and note that you did use the default settings, and if not, enter the settings you did use, because that will be super important to know what settings the recordings were captured under. And this year we have updated the field forms to provide suggestions. I don't have an image of that because this is just the full example, but we put a little bit of a gray number underneath this so it's clear what the default setting would be. If you're not completely familiar with it, you can just kind of note that it was default. Next, we talk about the temperature and RH logger that will be deployed in your grid cell, noting yes or no, it was deployed in this quadrant. And if it was deployed in this quadrant, fill out the other 3 boxes, i.e. the model of the temperature logger, and that's what I was talking about before, the Bluetooth hobo, the optical hobo, or the Bluetooth Kestrel. And what type of housing it's in. They should 99% or all of them be in radiation shields now. We have a couple older versions of plastic tubs with holes in them and stuff like that, but they should all be in radiation shields now. And if they're not, please note that because that'll be a very important thing to know. And then last, the serial number or ID of that temperature logger as well. And that's found on the device itself or on the ambient shield. Next, we talk about the deployment area details. So we're asking information like distance to clutter, and that's just straight up distance between your microphone and the nearest object, what type of object that is, and this will influence the recordings themselves. So a tree will produce a much more fuzzy reflection than a flat surface like a building or a lake would. And if we get reflections off a lake, it can sometimes look like there's a second bat in that recording when it's just an echo. New this year, we're also asking for a measure of volume of clutter around your microphone, and clutter being objects. Basically, we're asking you to estimate the percentage of a 50-meter radius sphere centering on your microphone that would be filled by objects. So it's a very rough estimate, but say if you imagine that your microphone is in the middle of a clearing in a forest that is, let's say, 20 meters in diameter, 10-meter radius, you would expect that that's, I don't know, roughly 80% of your detection volume is filled with trees, and the 20% that you're monitoring in there is what's free. So your clutter would be 80%. Habitat type, just a rough land class of where your detector is located in this quadrant. The description of any changes in local habitat since last year. This is particularly important for things like wildfires, but also major site changes like somebody built a building right next to me, or building burned down, building was knocked down, so on and so forth, or even trees have grown significantly since initial monitoring or something like that can be sometimes useful to note here as well. General description of deployment location, i.e. whether it's along a river edge or in a clearing of forest, so on and so forth. I typically take this field as a manual field I'll look at just to get a sense of what you're monitoring. Whether or not the detector is located in a flyway, we ask if you do try and make an attempt at answering this one, basically saying, is it sitting along a linear feature? Is there a likelihood that the bats in this area will be flying between site point A and point B as opposed to sitting here and circling around eating? So more or less, is it not deployed in foraging habitat or things like that? So if it's right next to a lake where you get a lot of insect activity, it is not in a flyway. But if it's along a road that would lead to a lake, that is a flyway. Feature sample is a forest clearing or whatever else. Distance to nearest bat roost. This was what Corey was talking about the integration with some of the information from BC Community Bat. Knowing where these bat roosts are in your grid cell is quite important. So if there is a roost known to you that is monitored especially nearby, please do note that, and note the distance that it is from your detector. If the roost is right next to our detector, we'd expect obviously a huge number of recordings relative to if the roost is 2 or 3 or 4 kilometers away. So this gives us a lot of information on what we should be expecting to see from your detector. And then if you do have information on the bat roost, noting the species can also be quite helpful too. So we can say, yes, we expected those little brown recordings, but we didn't expect the silver hair bat, or we didn't expect the hoary bat recordings to be so high in this grid cell still, despite that nearby little brown roost. Oftentimes the species of those roosts, but if you do some roost counts or guano collection, I believe the BC Community Bat can help with some genetic analyses. Mandy would be able to correct on that though. And last part of the field forms here, we ask for you to note the number of people you take out in the field with you, as well as their names, and the hours of labor that took you and your helpers to deploy and retrieve the detectors in your grid cell. This part down here, we're actually just using to tell our funders how broad the involvement in the program is, and just how much time that you and your volunteers and your helpers are contributing to this effort, and how broadly it reaches. And then quick yes or no of whether or not you've submitted your new photos from the year and signed a photo release, and any other comments that we haven't otherwise captured in this field form.
Jason, there's just been a couple of questions in the chat.
Yes.
I was going to relay those. Perhaps Aaron and Jason, you guys want to address. One question was just basic about the field forms themselves. Trying to find them on the Google Drive. Doesn't look like the new one is there, but presumably someone at the office can upload that.
Yeah, I can address that right after this call.
Okay, so that'll wait though. There's not one to load on there right now.
There is one to load on there right now, but it's on my laptop right now, kind of thing.
Yeah, no, that's okay. I didn't know if the Castle office had one. Okay, that's fine. And then the second thing is about percent clutter. So the question was there a height to consider? So just trees or tall shrubs? And then for the percent clutter measurement, like I see you've got, Aaron, I think this diagram come from you, very cute little hemisphere. And, it's a little too blurry to see what those numbers are showing. Is there a certain height that is being recommended there for clutter?
Yeah, we actually have a paired document that we've adapted from the, it was Northwestern Bat Hub, where we're following their procedures on this one. So I can send that around.
I think it was like 50 meters in kind of all directions. So just, kind of an estimate in a 50-meter surrounding area.
That's what I was hoping it showed.
I'm pretty sure those numbers say 50. So
yeah, they're the 50-meter radius is what those lines are indicating.
Yeah. Okay. No, that sounds good. Basically, I mean, some of the hoary bats and so on, you could be getting up to 100 meters away, but on average, most of the bats are going to be recorded in 50 meters. So that would be a good number to use roughly then for estimating your percent clutter away from the microphone. Thanks for asking that question, Juliet.
I just want to clarify though. So when you're looking in a 50-meter radius, what are you considering clutter? Just trees? Like anything above microphone height? For example, I'm in a boulder field in one of my sites. So I'm just curious, like how high does the clutter need to be considered clutter? Or maybe if you upload that document on how you measure it.
Yeah, I mean, the document has a little bit more details, but yeah, basically anything above the landscape that's, I would say, greater than a couple feet kind of thing. And it's a very rough estimate too. So small objects, you could estimate is like a couple percentage if it's just a couple small boulders in a field that's otherwise completely empty. Small objects should only be like 1 or 2% kind of thing too. So it's very just rough.
Okay, thank you.
Is it
And
fair enough to say, Jason, that basically if you kind of imagine you are the person kind of where that microphone is in that hemisphere, and you kind of estimate how full it is?
yeah,
Like the airspace, how full is that airspace?
yeah, exactly. And just, exactly. Basically visualizing what percentage of the airspace is filled by objects of any kind. Okay, I think
Sorry,
that
so
looks
it's
like
like
the
a
last question.
3D, like a 3D
Yeah.
kind of measurement?
Yes, just
So if you're in a boulder field, it might, and the boulders are like as tall as you are, that might be like 50%.
I would say a little bit more than that, because 50 meters above you will extend quite a bit further up. So the volumosphere that boulders that are as tall as you are would take up is probably more along the lines of like 20% of the space, I would think. But,
Right, okay.
but
Thanks.
yes, totally. That's a good way to visualize it, I think.
Yeah, hey Jason, it's Brian. I was just wondering, like, you know, because the word clutter, you know, we often think of it as something being quite close to the detector that's going to influence the recordings. You know, maybe that's just where a bit of the confusion is. If you're getting 30 meters out, then it's still, I guess it is still cluttered, or just clutter within that 50-meter radius is what we're trying to say here, I guess.
Yeah,
eh?
kind of more from the bat's perspective than from
Yeah.
the person's perspective. So like it might look like it's not cluttered to us, but it is still to a bat that would be flying down that area, kind of thing.
Right. Okay.
Yeah, I think, I mean, you could bring up a good point, Jason, or Brian. I think that, you know, the type of clutter you were referring to when you showed your picture was reflective clutter, right? So ultrasound was reflecting off of something close to the microphone and then bouncing into the microphone, in some cases in a very messy way. And so then that would create those echoes. So that's the clutter that's really close by, that's acting as a reflector and making messy recordings. And then there's the as you said, Jason, this bigger concept of clutter in from the perspective of the bat. And then as soon as the bat's within a couple of meters of that clutter, it's reacting differently and it's producing different calls. And so if you're recording a bat that's flying within detection of your microphone, which we can say then very crudely is roughly 50 meters in all directions from your microphone, then that bat has a chance that it's going to change its echolocation calls to be something that's not going to be representative of the species, like that you can easily, or in some cases easily tell the species based on their search phase calls. So let's say 20% of the sphere then is clutter. You know, you could roughly kind of think, well, there's, you know, maybe a 20% chance then that bat is going to produce some shit calls. And then you won't be able to ID the bat, right? Versus if you say like 90% of that sphere is something, there's leaves everywhere, there's branches, there's rocks, whatever, then there's a really high chance that what you're going to be recording there is actually not going to be easily identified to species. And that's kind of the concept for using some sort of estimate, just so that we have a sense of what is the chance that we should be able to get a species level ID from that detector for those files. Yeah. Thanks.
Yeah, thanks, Square. That was a great way to explain that, I think.
I'll just add to 50 meters is actually like a decent chunk out. So I know when I've done this before, like I've paced out what 50 meters looks like, and it's the percent clutter ends up, I think, a little bit. People might overestimate it. Just it's 50 meters up is quite a bit. So pacing it out, if you have that opportunity, just to get an idea of what 50 meters looks like, could be a worthwhile thing to do. So.
That's a great point, actually. Yeah, because 50 meters is not necessarily a unit that we're used to estimating ourselves, and pacing it out directly is a great idea. Okay, I think that's the last question. Okay,
Yeah,
we'll
I mean,
switch
if,
back to
if
the last.
anyone has more questions, put them in the chat or speak up, right? Or what do we have time for that right now, Jason, or do you have more?
I just have the Transect field forms and then survey 1, 2, 3 to go through, and then we have the formal question period kind of thing.
Okay, yeah, no problem. Just
Sounds good. So we'll just talk about the Transect field forms pretty quickly here. We're asking for very similar site information to what you just saw on the stationary field forms, except a little bit less detail, obviously, because you don't have as much information along the entire transect. So we're asking for you to list the grid cell ID. Again, GRTS ID also is same thing. A rough estimate of clutter along the transect. So this is even more rough than the clutter in that sphere around your microphone. This is just basically, were you driving through an entirely forested area, or were you driving through a forested area that then opened up into a nice flat, nice open area kind of thing? Entirely subjective, but if your entire transect is along a forest road with forest all around, you're going to say something like high clutter. Or if you do open up a little bit more, say something along the lines of medium clutter. We're just playing with this to see if it gives us any information on whether or not the gives us information on how high quality the recordings are along your transect. Next, note the bat detector model that you used. It's either in SD1 if it comes from a WCS kit, or the SD2. EM touches are used in the parks kits, and we have a couple Swifts around in a couple of specific grid cells. Write the bat detector ID, similar kind of thing. It should be written on the device itself or the serial number as well. That would be just fine. Microphone position, typically on the back of the truck roof or vehicle roof.
Jason, I'm not completely sure we're on the right slide.
Does it say reminders and updates on the top and shows half of a image that ends right here, kind of thing?
Maybe we are. Okay, just looked like the stationary one. Sorry.
Okay.
Okay.
Yeah, it's fairly similar visually.
Okay.
And this is the site information, so it is quite similar. I've got the nightly information on the next slide, chopped off the lower half of the sheet. Yeah, so then you write the serial number of the hobo that you're using attached to your car mount. That, as I was saying earlier, we're collecting information on the temperature and humidity along a transect each minute to get a little bit more precise estimate of that variable, that covariate. Settings on detectors, these are also default, and we also list a little gray default for you to just kind of enter that in. Gain setting dial is actually specifically for the SD2 or SD1 here, and that's that little dial on top of your device. It should be the same gain setting each year. So that's something that's very important to list in your field form here. GPS and car mount IDs also painted on the devices themselves. Microphone type, whether it's an NABat for the stainless for the NABat SD2 or an EM Touch itself is the microphone. Or the microphone itself contained in the unit, rather. And then your start and end latitude and longitude. We're asking for these again in decimal degrees because they're easiest for us to use. And then note the map datum that you use, whether it's NAD or WGS84. The lower half of the sheet for transects is this reminders and updates thing. It is, excuse me, it's a long presentation here. This lower half of your field form is a place to note the nightly information from your transects. So the date, the start time, the end time, your name, and names of people who are with you down there. The estimated cloud cover when you began, the influence of the moon. So these two are quite related. If the cloud cover is high and the moon is out, but completely behind clouds, you would say low influence of moon because the moon is covered and don't really get much light from it.
Sorry, Jason, I think we've confirmed that we don't see the slide you're talking about.
Oh, okay.
Yeah,
Maybe
sorry.
I'll stop the presentation and restart. What does it look like right now to you?
It's still the passive one with the sphere. Half
Oh,
sphere.
interesting.
Yeah.
Okay, yeah, sorry, I'm not sure why that's not moving through. I'll restart from current slide. And Okay, how about now? Does this look better?
Perfect.
Great, thank you. So I'll just quickly flip back. This is what the top half of the transect sheet looks like over here. And this is what the one side of the lower half will look like with the nightly information from that transect itself. I think I was down here talking about the influence of moon and cloud cover. So if cloud cover is high, your influence of moon will be low. Otherwise, if cloud cover is low, the influence of the moon is simply going to be something along the lines of how full was it and whether it was above or behind a mountain while you're driving your transect kind of thing. Was there a lot of light from the moon? You note the start and end temperature. This can be read from the anemometer in your kit. Otherwise, the in-dash temperature, as long as your car hasn't been sitting there, is usually fairly accurate, quite accurate too. Relative humidity coming from your anemometer. Start and end wind speeds, similar kind of thing from your anemometer. Note any precipitation that happened along the transect. Typically, we ask you to stop the transect if you get any precipitation and pull the equipment down, because rain on your transect actually ruins the electronics you're driving around. But in certain areas, rain is completely unavoidable because it could rain for an entire week and a half, just a little tiny bit. So if you're on the West Coast in one of those spots where it rains all day long, all week long, very light rain that doesn't pool on your windshield is okay, but do stop if it gets to be heavier than that. So just like, yeah, tiny drops that don't pool. And then otherwise, note the hours of labor per person on your transect. So one transect to drive out and drive back is about 3 hours for 2 people. And last, any comment about the transect, whether something we didn't account for in this field form comes up, please just note it there. It'll be a manual entry, manual read form or field. And last thing, but new this year that I wanted to talk about here is the field forms. We've adapted them into a Survey 123 field form or Survey 123 form. And Survey 123 is an Esri product that allows you to enter your field forms into your smartphone, tablet, or laptop. And the idea here is that hopefully this might be a little bit easier for some of you who like to carry around your tablets into the field. I know BC Parks has tablets they carry with them quite often, so we're trying to make things a little bit easier for you guys to enter this information so it's not so onerous. And I'm not going to go through the specific forms because all the information is the same as the field forms there, but basically it's a scrollable kind of image here. It looks exactly like this. You scroll down the sections and fill in the boxes each at a time. The information, again, being the same as what I've just talked about before. The only notable difference here is this location one. It will actually pull your location from where you are when you're entering the information if you let it. For instance, if your tablet has a GPS module in there or you're using your phone, you can just pull the location and you don't have to look up your latitude and longitude. It'll automatically take that. And otherwise, this field form option does allow us to do a little bit more validation that checks to make sure that you're entering the right information that we want, whether it's I want a numeric value for this or whatever else. It'll check that and tell you, no, you have to send it in as numbers. And yeah, accessible through tablets and smartphones. And I will have the link for you in a subsequent email to this as well. I'm just doing some final testing on the survey itself to make sure it's functioning as I expect it to and giving me the data like I expect it to. Okay, and I guess actual last thing is the reminders checklist. We have included this as of last year. In the front of your binder is the checklist of procedure to run through as you're deploying and retrieving your detectors. And then we also include there is also a laminated kit for people who don't receive their equipment from us directly. With that, I want to acknowledge all the organizations that are involved in collecting data for us each year, as well as all the individuals on this call and all the individuals not on this call, the grid leaders, the volunteers. This program is entirely supported by your efforts. It could not operate without the massive amount of time you guys contribute to collecting these recordings. And as well, I would like to acknowledge all of our funders. We are a very broad and diverse program funded by a lot of different organizations. So with that, are there any other comments and questions?
Hey, Jason, I'd just like to make the point that the binders did go out in the kits, but they're not necessarily up to date.
Okay.
We didn't have time to update things, so people should check on Google Drive just to make sure they're using the most up-to-date information.
Right, that makes sense. Thank you, Heather. Yeah. So all the most up-to-date information will always be on Google Drive as we pull it out. We'll be posting up there. So if anything doesn't quite make sense, do check Google Drive, please. All right.
And I'm just seeing a comment in the chat then, Jason from Juliet. Thank you for all your coordination and hard work to make this happen.
Well, thank you, Julia.
Yes.
Yeah, and thank you for your dedication. Like, you've been a good leader from the very beginning too. So
Yeah, well, and yeah, kudos to everybody. I mean, as you said, Jason, this is one of those huge team efforts. Like, I don't think I've ever been involved with a project that just has so many partners and collaborators, and it's just, you know, it's massive and it's working. That's the cool thing is, you know, you start to get programs this large, and it's really all about coordination. And so yeah, so thank you for shepherding everybody through this process, and for the whole team, because yeah, this really is a team effort for everybody, including right down to the people deploying at the grid cells, all of you, and all the way up to our advisory committees. You know, Mandy, I see, is on the call. She's been very helpful, very instrumental in coordinating this program, as you mentioned as well, Jason, and lots of other people that, yeah, that this couldn't happen without. So
totally, yeah.
everybody give yourself like a pat on the back. There you go. Good job.
Definitely. And I mean, I mentioned a bunch of times we do prepare for like grid leader succession and grid leader turnover and stuff like that, but the amount we have has been so small that I am super impressed with just how dedicated everybody has been. Like, most of our grid leaders have been around for 5+ years kind of thing. So it's great to see how much consistency people give towards this project.
And hopefully will continue for many years. And even though we do want to change the role that we're playing in this program for reasons, obviously, you know, Jason pointed out, there are a lot of other knowledge gaps and threats and policy concerns and a lot of priorities that we hope to shift some of our energy to over coming years. So it seemed like a natural progression for us now, this program sitting where it is, to hopefully be able to have other people, other agencies or organizations help kind of with the heavy lift to coordinate this. So yeah, we look forward to continuing NABat, but in a slightly different format from what we have been doing up till the end of year 8, which you guys obviously are heading into right now. So have a fun year or fun summer, and good luck with all of your deployments.
Thank you. And any questions, just let us know at any point, anytime. We're happy to answer.